Chronic stress does not just feel bad, it ages you at the cellular level in a way that is measurable, documented, and directly connected to the biological mechanisms that determine how long your cells can continue to divide and function. The mechanism is telomeres, and understanding what telomeres are, what shortens them, and what slows that shortening changes how you think about stress from an emotional problem into a biological one with specific, measurable consequences. In 2009, the Nobel Prize in Physiology or Medicine was awarded to Elizabeth Blackburn, Carol Greider, and Jack Szostak for the discovery of how chromosomes are protected by telomeres and the enzyme telomerase. Their work established the molecular basis for cellular aging and inadvertently created the most precise biological clock for measuring the impact of lifestyle on the aging process. Every chromosome and every cell in your body ends with a structure called a telomere, a repetitive DNA sequence that caps the end of the chromosome the way a plastic tip caps the end of a shoelace. The sequence is the same in every organism that has telomeres, TTAGGG, repeated thousands of times. Telomeres serve a specific structural function. DNA replication machinery cannot copy the very end of a linear chromosome. It needs a primer sequence upstream of the region it is copying, which means the final segment is always lost. Without telomeres, this loss would occur in the protein-coding regions of the chromosome, progressively deleting functional genes with every cell division. Telomeres sacrifice themselves instead. Each time a cell divides, the telomeres shorten by approximately 50 to 200 base pairs. The protein-coding DNA is protected. The telomere absorbs the loss. This continues until the telomeres reach a critical minimum length, called the Hayflick limit, at which point the cell can no longer divide safely. The cell enters a state called senescence. It stops dividing but remains metabolically active, secreting inflammatory signals that damage surrounding tissue. Or it undergoes programmed cell death. Either outcome reduces the functional cell population in the tissue. Telomere length is therefore a direct measure of cellular age and remaining replicative capacity. Cells with long telomeres have more divisions remaining. Cells with short telomeres are approaching or at their replicative limit. Tissues composed of cells with shorter average telomere length are biologically older regardless of their chronological age. This is why telomere length has emerged as one of the most informative biomarkers of biological age. It integrates the cumulative history of everything that has caused cellular stress and accelerated division throughout a person's life. Blackburn's research, conducted with her graduate student Carol Greider, established not just the structure of telomeres, but the existence of telomerase, the enzyme that can rebuild telomere length. Telomerase adds TTAGGG sequences back to chromosome ends, partially reversing the shortening that occurs with division. Most somatic cells, the cells of adult tissues, have very low telomerase activity. They shorten with each division and do not rebuild. Stem cells, immune cells, and certain other specialized cell populations have higher telomerase activity that partially compensates for division-related shortening. Cancer cells typically have very high telomerase activity, which contributes to their unlimited replicative capacity. The existence of telomerase established that telomere shortening is not inevitable at the rate it occurs under normal cellular conditions. It is a regulated process that can be modulated. The discovery opened the question of what factors accelerate or decelerate telomere shortening beyond the baseline rate of cell division. The answer involves oxidative stress, inflammation, and, most relevantly for the question of chronic psychological stress, cortisol. Cortisol is the primary glucocorticoid stress hormone. It is produced by the adrenal glands in response to activation of the hypothalamic pituitary adrenal axis, the hormonal cascade that the brain initiates in response to perceived threat. In acute stress responses, cortisol is functional and necessary. It mobilizes glucose, suppresses immune function temporarily to redirect energy toward the immediate threat response, and prepares the body for physical action. The cortisol spike during acute stress resolves when the stressor resolves, and the body returns to baseline. Chronic stress, sustained psychological stress from ongoing life circumstances, produces sustained cortisol elevation rather than resolved spikes. This sustained elevation has multiple documented biological consequences, but its effect on telomeres is among the most directly measurable. Cortisol inhibits telomerase activity. Research has shown that glucocorticoid exposure, either from stress or from pharmaceutical glucocorticoids like prednisone, reduces telomerase activity in immune cells and other dividing cell populations. Without adequate telomerase activity to partially compensate for division-related shortening, telomeres shorten faster. Cortisol also promotes oxidative stress. It shifts cellular metabolism in ways that increase the production of reactive oxygen species. Oxidative damage to telomeric DNA accelerates shortening because the telomeric sequence TTAGGG is particularly vulnerable to oxidative modification, and oxidatively damaged telomere bases cannot be replicated normally, producing additional length loss at each division. The combined effect, telomerase inhibition and increased oxidative damage, means that chronically stressed individuals show measurably shorter telomeres than age-matched controls who are not under chronic stress. This is not theoretical. It is directly measured. Blackburn's research group conducted a landmark study comparing telomere length in mothers caring for chronically ill children, a population under sustained, objectively verifiable chronic stress, with control mothers of healthy children. The caregiving mothers had significantly shorter telomeres. Among the caregiving mothers, the length of time spent caregiving correlated with the degree of telomere shortening. The women who had been caregiving longest had the shortest telomeres, corresponding to an estimated additional 10 years of cellular aging compared to controls. 10 years of additional cellular aging from the cumulative biological effect of sustained psychological stress. This is the mechanism behind the clinical observation that chronically stressed people age faster and die younger, not as a correlation, but as a causal chain from stress hormone to enzyme inhibition to accelerated telomere erosion to reduced cellular lifespan. The interventions that slow telomere shortening follow directly from understanding the mechanism. Aerobic exercise is the most consistently documented telomere protective intervention. Multiple studies have found that regular aerobic exercise is associated with longer telomeres in adults of the same chronological age, with effects that appear to be dose-dependent up to moderate training volumes. The mechanisms involve reduced oxidative stress, improved antioxidant defense systems, and increased telomerase activity in immune cells following exercise. A study comparing endurance athletes to sedentary age-matched controls found telomere lengths corresponding to approximately 9 years younger biological age in the athletes. Meditation and mindfulness practices have documented effects on telomere length through mechanisms involving cortisol reduction and inflammation reduction. Blackburn herself conducted research on the effects of meditation retreat on telomerase activity, finding that participants showed significantly higher telomerase activity following intensive mindfulness practice compared to controls. Higher telomerase activity corresponds to more effective telomere maintenance. The Ornish program, a comprehensive lifestyle intervention combining plant-based diet, moderate aerobic exercise, stress management through yoga and meditation, and social support, produced measurable increases in telomerase activity in prostate cancer patients in a pilot study, with longer follow-up data suggesting actual telomere lengthening. This is one of the few interventions that has demonstrated not just slowed shortening, but actual reversal of telomere length loss. Sleep quality has direct effects on telomere length through cortisol regulation and inflammatory pathways. Poor sleep elevates cortisol and inflammatory cytokines, both of which accelerate telomere shortening. Studies have found associations between short sleep duration, poor sleep quality, and shorter telomeres, with the relationship independent of other lifestyle factors. Chronic psychological stress reduction, through whatever mechanism is effective for the individual, directly addresses the cortisol telomerase inhibition pathway. The specific method matters less than the effectiveness in reducing sustained cortisol elevation. Exercise, meditation, social connection, therapy, and changes to the stressful circumstances themselves all address the same upstream mechanism. Omega-3 fatty acids, EPA and DHA from fatty fish, have shown associations with telomere length in prospective research. A study published in JAMA found that higher omega-3 levels in cardiovascular disease patients were associated with slower telomere shortening over 5 years, with the relationship dose-dependent across the range of omega-3 levels measured. Vitamin D sufficiency is associated with longer telomeres in population studies, with proposed mechanisms involving reduced inflammation and improved DNA damage response. Given that vitamin D deficiency is widespread in populations with limited sun exposure, this association has practical relevance for a large proportion of the population. Here is the synthesis. Telomeres are the molecular clock of cellular aging. They shorten with each cell division, and when they reach critical minimum length, the cell can no longer divide, entering senescence or death. Telomere length integrates the cumulative biological history of stress, inflammation, oxidative damage, and division throughout life. Chronic psychological stress accelerates telomere shortening through 2 documented mechanisms: cortisol inhibition of telomerase, which reduces the enzyme that partially rebuilds telomere length, and cortisol-driven oxidative stress, which damages the telomeric DNA sequence that is particularly vulnerable to oxidation. Blackburn's research established that the biological effect of chronic stress on telomeres is measurable and substantial, up to 10 years of additional cellular aging in chronically stressed caregivers compared to controls. The interventions that slow shortening address the mechanisms directly: aerobic exercise, 9 years younger telomere age in endurance athletes; meditation, increased telomerase activity; omega-3 fatty acids, slower shortening over 5 years in prospective research; sleep quality, reduced cortisol and inflammatory telomere damage; stress reduction through any effective method, reduced cortisol, reduced telomerase inhibition. Chronic stress is not just a psychological state, it is a biological process that shortens the molecular structures that determine how long your cells can function. The Nobel Prize was awarded for understanding that mechanism. The interventions that protect against it are not complicated. They are exercise, sleep, omega-3, meditation, and reducing the sustained stressors that keep cortisol elevated. If this changed how you think about stress as a health variable rather than just an emotional one, hit the like button. Share it with someone who thinks stress is just part of life, and subscribe for more content like this.